Hello, this is Milton Greenberg. Welcome to Microbiome and Mucosal Immunity, the next lecture in our medical immunology course. Today we will introduce the mucosal surfaces and the immune composition. We'll discuss and introduce the microbiome in the context of immunology. We'll discuss leukocyte trafficking and homing, specifically focusing on chemokines and integrins. And we'll discuss different mechanisms of pathogen clearance. And we'll introduce the immunopathology of chronic colitis. So the mucosal surfaces throughout our body have 200 times more surface area than that of the skin. So you can see the mucosal surfaces of the body. This includes your respiratory tract, your gastrointestinal tract, mammary glands, and the urogenital tract. So our physiology and immunology are strongly influenced by the environment. So these mucosal tissues contain immune cells that are dynamically interacting with the environment. And this includes commensal microbes that live on us, as well as pathogens that we might encounter in the environment. And then also these will work together to determine the ultimate health outcomes. So the reason this is important is that worldwide deaths annually from mucosal infections, particularly acute respiratory infections, are very high. And so mucosal infections globally are a major health problem. In the United States, this was less appreciated because of our strong disease control and prevention measures. However, with the onset of the COVID-19 pandemic, this has all kind of brought this back, and we've really redoubled our focus looking at the specific immunology and the interaction between microbes and pathogens and the mucosal immune system. So these mucosal infections can be therapeutically managed. We've had a number of advances in antivirals in the case of acute respiratory infections, but diarrheal diseases, HIV/AIDS, is that all of these will be coming back both in this class and future microbiology classes as we learn more about these pathogens and their specific vulnerabilities. So there are more microbial cells on our body living in us and on our surfaces than there are human cells. By mass, the human cells are more because the microbial cells are much smaller, but so many different microbes compose our microbiome. And so these are different classes of different phyla of different microbes that live inside our body. And so everybody's mucosal fingerprint is very different. It's that the microbes that live on you and with us and help regulate our immune system, help supply vitamins, help protect us from pathogens that might want to colonize, is that all of these are going to be slightly different person to person, giving everyone our unique microbiome fingerprint. And furthermore, is that these are highly regulated by what we do to our own bodies. So this could be circadian regulation, so different times of day, the mucosal microbiome will shift. Also, depending on what we eat and our nutrition, is that these microbes will shift, and microbes that like what we eat will start growing, and microbes that like other diets will kind of go away. And then also in different disease states. So when we'll go through a couple examples of when certain pathogens take up residence, is that they can start overcoming the natural microbiome that we have, the commensal microbiome that is providing benefit to us, and start dominating and causing different disease states. So looking at the different phyla of microbes, is that there are many different phyla that live in different parts of the body. So in the gastrointestinal tract, you can see it's really dominated by firmicutes. In the skin, it's really large population of actinobacteria. And again, you don't need to memorize all of these. It's just showing the diversity and how many different phyla we see and how many different microbes there are at different regions of the body, and then dynamic interactions that they play with our host physiology as well as the immunology and how the immune response can respond to these microbes. So we have diverse features of the mucosal immune system. So we have anatomical features that are really unique. We have different effector mechanisms, and we have this immunoregulatory environment. So this is taken directly from Janeway. If you want to read more about this, please look at Janeway. And then also, if you have minimal experience with any of these topics, please go back to the textbook and start reading and understanding more about how this works. Again, how the immune system works is a really good primer, very quick read that can help reinforce these basic concepts that can help us moving forward. So these intimate interactions between the mucosal epithelia and the lymphoid tissues, we'll be discussing this a little further in today's lecture when we talk about immune cell migration. And then we have discrete compartments. So compartments are areas that immune cells like to hang out. So these could be structures like Peyer's patches. Peyer's patches are unique structures in the gut that help maintain the immune cells so that they have ready surveillance of food and other microbes that might be transiting through the gastrointestinal system. We have isolated lymphoid follicles, we have tonsils. So these are some secondary and tertiary lymphoid structures that are just anatomically present adjacent to our mucosal surfaces. We have specialized uptake mechanisms, and this means that of all these things that are transiting our body, for example, in the gastrointestinal tract, is that you're putting things into your mouth. And so the immune system has special ways to sample the antigens where it can understand a little bit more about what is trafficking. Is it something good? Is it something bad? And so we have M cells that we'll be introducing. We have these Peyer's patches, adenoids, tonsils, and these M cells can help maintain this antigen uptake. So we have activated and memory immune cells that can dominate and predominate even in the absence of infection. We have multiple activated natural effector and regulatory T cells present. What do I mean by this? Is that inside our gut is that there's so many microbes, and these microbes have molecular patterns that are there to activate immune cells. And these immune cells can be activated, and they can be activated in certain ways, depending on exactly what is happening with the entire system. We have antibodies, secretory IgA antibodies that are very effective at dissolving in these mucosal tissues. And we have distinctive microbiota, and these microbiota, again, can help prevent the colonization of pathogens. We have an immunoregulatory environment. What this means is that we predominantly down-regulate these pro-inflammatory immune responses. So we're not going to have immune responses to food or other innocuous antigens. In a typical person is that you can eat and you won't develop an immune response. So even though that there's foreign things going through the system, you won't have a strong pro-inflammatory response in a healthy individual. And this is mediated by tolerance. So we have inhibitory macrophages, tolerance-inducing dendritic cells, things that are presenting antigen in the absence of pro-inflammatory cues that can set off a really strong pro-inflammatory immune reaction. So the immune system can distinguish these commensal microflora from the harmful pathogens. So these good bugs that live in us versus these bad bugs that seek to do us harm. So these intestinal lymphocytes are found in these organized structures. So we have in the colon, for example, in the intestine, is that we have these these linings. These are going to be called epithelial cells, and depending on where you are, these could be enterocytes or colonocytes, and they're forming these invaginations that increase the surface area, help with nutrition and nutrient and water absorption. And within here, we have a number of immune cells. So we have intraepithelial lymphocytes that will live directly inside the epithelium. And then this deeper tissue is called the lamina propria. The lamina propria has a ton of immune cells as well. And these immune cells are vigilant and they're sitting there surveilling the tissue, understanding what's happening, understanding what's going through the tissues. And from this is that they're kind of on standby, ready to activate in a pro-inflammatory context if needed. We also have lymphatics, and these lymphatics are helping with the absorption of fats, but also sending signals out to the mesenteric lymph node. We have organized lymphoid structures like these Peyer's patches. These Peyer's patches will help with these organize T cells and B cells into their respective areas, so you can have this nice concerted and highly regulated activation of B cells and T cells. And this way the B cells and T cells don't have to migrate very far. But we also have the mesenteric lymph node that is a short distance away as well. So these organized lymphoid structures help regulate the commensal immunity. Organized lymphocytes will dynamically interact with these mucosal barriers to help protect us from bad bugs. So who lives in these tissues? Is that all of our favorite cells. So we have CD8 positive T cells. So the mucosal immune system consists of different compartments. First in the epithelium, which is the outer lining that separates the lumen from our cells. And then also the lamina propria, which is this deeper area here. So most of the immune cells are in the lamina propria. And this is where you have a number of T cells, both CD4 and CD8, antibody secreting plasma cells that secrete IgA. IgA dissolves very well in mucosal tissues. You have mast cells that can help release histamine and facilitate allergic reactions. So these immune cells of the lamina propria, you can see them here, is that they have specific receptors that mediates where they end up. So in particular, CCR9 is a chemokine receptor that helps get these cells to the gut. And then once they're in the gut, there's different integrins. And these integrins are adhesion molecules on the surface of these cells that help them migrate to areas where they're supposed to be. So for example, the alpha-4 beta-7 integrin helps cells migrate through the lamina propria, where the alpha-E beta-7 integrin helps cells migrate through these epithelial layers. You can also see you have this dendritic cell that kind of spans both sides. So this dendritic cell is reaching out a dendrite across the epithelium, and it's actively sampling antigens that are coming through that luminal side and carrying them back into the lamina propria so it can discuss what's happening with the T cell and other immune cell buddies that live in the deeper tissue. So these T cells home from the Peyer's patches, they home to the Peyer's patches from the blood. So here we have our high endothelial venules. So this high endothelial venule, this is a vein, this is a specific type of vein that the leukocytes can traffic, and they have specific receptors. Again, so here are selectins that help facilitate that rolling on the endothelial surface. We also have chemokine receptors that'll help these cells migrate to where they need to be. So the T cells can enter the Peyer's patches from the blood vessels, directed based off of the receptors that they have. So the cells that express the receptors that are important for entering these tissues will end up in the tissues. The cells that don't express these receptors will end up where they're supposed to be based off of their receptor expression. So here we have the cell in orange called an M cell. M cell are special mucosal cells that help with the trafficking of antigens. So we have antigens on the luminal side of our gastrointestinal system. And what happens is these antigens can come down, get presented by dendritic cells, and then these T cells can now get activated because now they're where they need to be. They have a T cell receptor that can recognize to those specific antigens presented in the context of that MHC, and therefore that these T cells can get activated. These T cells can then drain to the mesenteric lymph nodes through the thoracic duct and return to the gut via the bloodstream. So you're getting activation of these immune cells, and then they can migrate from the Peyer's patches and into the tissue. Here in the small intestine, we have the lamina propria. Here is that they're migrating into the lamina propria, and then they can migrate into the intestinal epithelium as well, depending on the receptors that they express. So these Peyer's patches, again, are small organized lymphoid structures, and homing to the gut is regulated by our integrins and chemokines that are all very important. So how does this work? We've learned a little bit about this earlier in the class, but this is mediated by rolling. So the cells can extravasate from the lumen of the blood vessels, and so they can start rolling the interactions with their selectins on the so the cells, the immune cells that are migrating, they will start interacting with the selectins, and this will help facilitate the rolling onto the endothelial cell surface. From there, they'll start sensing cytokines and chemokines. These cytokines and chemokines will start activating these immune cells, and these immune cells will become active. They'll become more sticky. The integrins will start changing their conformation to allow them to have a higher binding affinity to the adhesion molecules that are expressed on the different cells. And then this will mediate firm adhesion that is driven by these integrins. Here, this is an integrin LFA1, the linker for activated T cells 1. And then you're going to get diapedesis. Diapedesis is that extravasation process where the cell can start migrating between the gaps in between these endothelial cells and migrate from the luminal side of the vasculature into the tissue. So this is important, again, in the gut. So here is the blood vessel. And what we have is we have integrins, and these integrins here, this is a alpha-4 beta-7 integrin. This is binding to the cellular adhesion molecule, the mucosal CAM cell adhesion molecule. So this is one of the mucosal cell adhesion molecules that's expressed here. And so this cell is supposed to be in the gut because he or she expresses these specific integrins, and it'll bind to that adhesion molecule, make its way into the lamina propria, and depending on what receptor it has an affinity for, CCR10 will start homing to the large intestine, the colon, and cells that express CCR9 will start binding to a different chemokine and migrate their way to the small intestine. So gut homing effector T cells can bind to the endothelium, get where they need to go, and then depending on the chemokines and chemokine receptor expression, is that they'll start migrating either towards a small or large intestine. And the idea here is that a lot of organs express their own unique chemokines, and this helps traffic immune cells to where they need to go. This is also important for cancer metastasis, where as cancer cells begin migrating through the body, they will also upregulate some of these chemokine receptors to help facilitate their trafficking to different organs. So many organs express their own specific adhesion molecules, and this is important for organ-specific therapeutic targeting. And we'll get into this with the example of natalizumab. Natalizumab is a fantastic drug. It works very well for people that have multiple sclerosis. Multiple sclerosis is a chronic demyelinating disease of the central nervous system. So in this case, you have immune cells migrating from the blood vessels and migrating into the brain. And because of this, these immune cells that are normally not in the brain are now migrating into the brain, and they have activity for brain proteins and can cause brain damage as is what happens in the disease multiple sclerosis. So these integrins are good drug targets for tissue-specific immunosuppression. So the first drug that was identified here is natalizumab, brand name Tysabri. And just Tysabri works by preventing immune cells from migrating into the tissues. The way it works is that normally this alpha-4 beta-1 integrin will bind to the vascular cell adhesion molecule, the VCAM, and what will happen is that'll help T cells and other immune cells migrate their way into the brain or even into the gut. And in this case, if you're blocking these integrins with a therapeutic monoclonal antibody, natalizumab, that natalizumab will bind to the integrins. The integrins are no longer able to bind to the cell adhesion molecule, and therefore you're not getting immune cell migration into the brain. So if you look at this in an untreated patient with multiple sclerosis, they're getting more and more immune cells into their brain, more and more lesions identified by MRI, so a diagnostic technique used in this disease, and this just goes up and up, the disease progresses. But with natalizumab, which is an alpha-4 anti-alpha-4 integrin therapy, is that essentially the disease is stopped. For the majority of patients, the disease does not get any worse. So this is just a breakthrough therapy that really changed the lives of multiple sclerosis patients, particularly multiple sclerosis patients that were refractory to the standards of care. This was also approved for Crohn's disease in 2008. But then coming along is that, just really fantastic immunologists, pharmacists, gastroenterologists work to develop this vedolizumab. And vedolizumab specifically blocks T cell migration into the colon. So the vedolizumab inhibits interaction of a specific integrin called alpha-4 beta-7. And alpha-4 beta-7 is the one that binds to the MADCAM, which is that mucosal cell adhesion molecule. And then this was approved for ulcerative colitis and Crohn's disease, and is a great wonder therapy for people with ulcerative colitis and Crohn's disease. So just showing how important this immune cell trafficking from the blood into the tissues is. So here we're looking at ulcerative colitis. So in ulcerative colitis, you have a number of changes in the tissues that are driven by chronic inflammation. And we'll be studying ulcerative colitis a little more later in this class. But the idea here is that in ulcerative colitis, you're getting this massive growth, this massive hyperplasia, this massive hyperproliferation of the colonic epithelium. And here you can see just the standard histology from a normal, happy, healthy individual. You have these nice crypts, and these crypts are very important for absorption of water and nutrients. Here you have these little kind of like little white spots in the histology. Those are goblet cells that are making mucus. That mucus really helps the stool traffic nicely through the colon. And again, very kind of nice, tightly organized tissue. But in colitis, with this histology image, you can see that this is massively grown as you have a huge expansion and hyperproliferation of what's happening inside the tissue. An ulcer, you have an ulcer that forms, and this is what it looks like under the microscope. And you see there's no intelligent architecture here. All these crypts that we were describing are totally gone. These little purple cells, black dots that you see throughout the tissue, those are infiltrating leukocytes that are staying with that hematoxylin dye. So there's just so many immune cells in here, and they're all spitting out inflammatory cytokines, which is causing just very, very painful and very uncomfortable for a number of patients. So how does this work? It's a very complex slide. But on the left side, we have the healthy state. On the right side, we have this ulcerative colitis. And then here in green, these are approved drugs for ulcerative colitis. And then in purple are other drugs Some of these are now approved. At the time of the publication, these were investigational drugs that are being explored. And we'll learn more about these S1P modulators coming up later in the class, as well as tofacitinib is now approved for this indication. So let's kind of walk through this. So on the left side, we have a nice little Peyer's patch, a little lymphoid aggregate that has a number of T cells and plasma cells and a number of dendritic cells. There's a lot of this IgA, a lot of this mucus shown in yellow. And what this is doing is this is sampling the normal microflora, just making sure everything is okay. And here, this is our local mesenteric lymph node. We have mature dendritic cells. We have different immune cells that can then traffic from the local sites into the mesenteric lymph node and then back into the bloodstream and then back again. But in the case of mucosal injury and inflammation, there's a dysbiotic microflora. So you have other microbes that are dominating where they don't normally dominate in a healthy person. You have a number of different immune cells that are chronically inflamed and making all sorts of pro-inflammatory cytokines. In the intestinal lamina propria, you have, again, production of lots of different cytokines. You have infiltration of immune cells into that lamina propria. And so in order to stop this, one is you could use S1P blockers to keep cells in the lymph node so they can't migrate out of the lymph node and then cause damage in the colon. We have vedolizumab, etrolizumab. So these are your Tysabri and Tivio that can block the migration of immune cells from the bloodstream and into the tissues. You have anti-cytokine agents, things like infliximab, adalimumab. These are anti-TNF blockers that can block TNF, thus reducing the cytokine load and then also reducing inflammation. You have tofacitinib. Tofacitinib is a Janus kinase inhibitor, a JAK inhibitor that works inside the immune cell to stop that pro-inflammatory immune signaling. So here is just kind of a busy figure, but just showing in the natural state, you have all these immune cells, but then over here on the pathogenic state, you have a total dysbiosis, and then also the immune compartment is very out of whack, and you have lots of cytokine production, a lot of immune cells that are normally not inside the tissues, or if they are inside the tissues, are now chronically activated in a pro-inflammatory manner that is driving immunopathology. So let's take a little bit and learn a little bit more about how these antigens are passed through the intestinal epithelium. So we talked about the microbes that live on the surface. So the microbes can live in the luminal surface, luminal side of the gastrointestinal tract. And here we have our enterocytes and our M cells. So the M cells are helping microbes get through, antigens get through, where they can then be processed by the dendritic cells. We also have specific antigens that might be taken up by the enterocytes and trafficked across the barrier. We also have antibody-mediated transport, so there's a lot of IgA antibody in the mucosal surface. These are sampling microbes. This might be transported by these antibody receptors called Fc receptors to the dendritic cell. We also have this apoptosis-dependent transfer. So if there's dying cells, for example, if a cell's old and is dying, or if there's a pathogen that's killing that cell, that might cause some trafficking of these antigens to that dendritic cell on the lamina propria side of the tissue. We also have a couple other mechanisms. So on the left is, or on the top is a cartoon, and on the bottom is the image from the microscope that was used to draw that cartoon. So here in this white line, this is the enterocytes that line the intestine. And here you can see that these macrophages and dendritic cells are extending their dendrites out through in between these specific enterocytes, and they're kind of reaching through and sampling antigens that are coming through, and then they can bring those antigens in and present them and kind of feed them off to dendritic cells. Here we also have our goblet cells. So goblet cells secrete mucus, but they can also help with antigen uptake. And then we also capture by these intraepithelial dendritic cells. So some dendritic cells can reside in the tissue and they can capture things inside the lumen and then bring them back into the lamina propria. So these are just different ways in which the immune cells monitor the gut microflora. So this IgA is secreted by this IgA secreting cell. So IgA is very important because, again, it dissolves very well in that mucus, dissolves very well in that mucosal immune system, and it really helps with just sampling and monitoring of what's happening in these tissues. So here the binding of IgA is that IgA is secreted in the lamina propria. Then it binds a transport molecule. It's then endocytosed and transcytosed to the apical face of the epithelial cell, and then released out so it can start sampling and binding different things that are trafficking through the luminal side of the gastrointestinal system. We also have our CD8 positive T cells. So CD8 positive T cells can help kill virally infected epithelial cells. So when a virus comes in and starts binding and reproducing in these epithelial cells, what's going to happen is that this epithelial cell will notice that and it'll start presenting antigen, viral antigen in the context of MHC class 1. This will trigger CD8 positive T cells, and these CD8 positive T cells have T cell receptors that can interact with viral antigen presented in the context of MHC class 1 and secrete out its cytolytic molecules, perforin and granzyme, that can help kill these pathogenic viruses, and these viruses that have infected your cells, is that it can start secreting this out and start killing those virally infected cells. So here, this is just a list of all the different intestinal pathogens and their associated human diseases. So particularly in the winter months, is that these are a major issue as far as infectious disease is concerned. You don't need to memorize these, but it's just showing the diversity and kind of why there's so many different immune system mechanisms that are helping to monitor what's happening in the mucosal tissues, is because we're susceptible to salmonella, pathogenic E. coli, Clostridium. We'll go through some different examples of this, as well as viruses, things like rotaviruses, norovirus, adenoviruses. These can all cause, vomiting diseases. And, these are particularly pronounced in pediatric patients. Also, parasites. We have a number of different parasites that we're susceptible to that can come in through the intestinal system. So most parasites are bad, but again, you don't need to know all those for this exam. This is just kind of setting it up for your future microbiology class. So here, pathogen clearance, just general pathogen clearance inside the tissues. So you have all these microbes that live on the surface of your immune cells, on the surface of this mucosal immune system and your mucosal surfaces. How come you're not chronically having inflammation? It's because of what is happening there, is that most of these immune cells are most of these microbes are kind of staying in their lane. They're staying where they're supposed to be. And once these microbes start coming inside your cells, is that there's certain microbes that like to infiltrate inside your cells, or there's certain microbes that have these secretory systems where they can release toxins into the cells. And these can be recognized by toll-like receptors or nod receptors. These are pattern recognition receptors, PRRs. And so when there's things going on inside these enterocytes, inside these colonocytes, they will know that, hey, there's not supposed to be microbial stuff inside of me. Normally I'm like trafficking things, doing it in a very regulated manner. But if there's things that are in here and triggering my receptors, this is not a good thing. And because of that, I can activate NF-kappa B. NF-kappa B is going to go and get activated, go into the nucleus, start making a number of pro-inflammatory cytokines. These pro-inflammatory cytokines, things like IL-1, IL-8, CCL1, CCL2, these different pro-inflammatory cytokines and chemokines, these inherently antimicrobial proteins like defensins, all of these will start activating a number of inflammatory mediators. These will then help recruit and activate neutrophils, macrophages, dendritic cells. All those inflammatory cells will start migrating into the tissue to help kill the bad stuff, facilitate pathogen clearance. So let's give an example. So for salmonella, salmonella loves to enter through these M cells. They can also enter through these epithelial cells, enter directly into the epithelial cells and migrate through. They can also enter these phagocytic cells, so these macrophages or dendritic cells that are extending processes out into the lumen. They can bind wholly, enter the cells, and then migrate to the lamina propria. Once they're in the lamina propria, all those cells in the lamina propria will get activated. They'll start spitting out cytokines and chemokines. These will then help recruit in neutrophils and other immune cells from the blood into the tissues, and all of these will help causing the clearance of the salmonella inside the tissue. You're going to get activation of trafficking of dendritic cells into the mesenteric lymph node. Inside the mesenteric lymph node, you're going to start driving that adaptive immune response. You're going to activate T cells, activate B cells, start producing antibodies, getting those cells to traffic back into the gut to help with that pathogen clearance. And so this is what happens with a lot of people when they get salmonella infections, is there's just going to be a massive inflammatory response. It's going to be associated with inflammation and vomiting and diarrhea. But in some people that can't clear it in the colon, the salmonella, it's really good at trafficking, will eventually make its way into the bloodstream. If it enters the bloodstream, the survival prognosis goes way down because now you have a systemic infection, and this is giving rise to potentially a risk for systemic inflammation and potential risk for septic-like events, so septic shock, or some of these chronic bacteremia that you can see in this salmonella infection. So let's go through another example. So this is an example called Shigella. So in the case of Shigella, it has a more different pathogenic mechanism, but a lot of the same key components. So the M cells will help allow the Shigella to penetrate through the epithelial layer and into the lamina propria. Once it's in the lamina propria, the Shigella will migrate in through the basal surface of these epithelial cells and then start spreading transversely through these other epithelial cells. And then in here, they're going to start activating a number of pro-inflammatory mechanisms. So this will be your NF-kappa B, is because we have these receptors, these pattern recognition receptors inside the cell. These pattern recognition receptors inside the cell, the nod receptors, what this will do is this will activate NF-kappa B. NF-kappa B will go into that nucleus and start making a number of pro-inflammatory cytokines like CXCL8, IL-8, and that's a very potent pro-inflammatory cytokine and chemokine that's going to help drag in neutrophils into the tissue to help cause inflammation, and then those neutrophils can gobble up any bugs that are still in the lamina propria and really help with that pathogen clearance. Okay, so another example, this is a Clostridium difficile. So Clostridium can happen secondarily to antibiotics. So in this case, normal commensals provide a nice barrier to help prevent Clostridium from taking hold. But in a normal case, antibiotic regimen that you might have for like an ear infection, you're going to kill a lot of commensals in your gut. And as such, is that Clostridium might take hold, is that there could be Clostridium in you already, and that Clostridium wasn't killed by the bugs or by the antibiotics, and now that Clostridium has a wide open range to start proliferating and dividing, this can gain a foothold. Clostridium loves to secrete toxins that can cause mucosal injury. As these cells die, more and more immune cells will get activated, migrate into the gut, migrate into the tissue where you get this leaky epithelium caused by these dead and dying cells and injured epithelial cells. And this will lead to colitis, so inflammation of the colon and formation of this pseudomembrane, where you just have so many microbes growing and so many immune cells. And a lot of times this Clostridium are very good at fighting off the immune system through mechanisms that you'll learn in future classes. Thank you.